Welcome everyone to Cell Structure and Function Part 2. In this session we're going to be looking at the eukaryotes and why they are different to the prokaryotes. So we've looked at prokaryotes and you remember they're the bacteria, but eukaryotes are the more complex cells. Bless you. And so we're going to look at some structure and function. We're going to look at some of the internal organelles as well, and also, so inside the cell, but also outside the cell, because if you remember your Nat and Phys, that extracellular matrix is quite important as well for signalling on the cell. We're also going to look at a little bit of endosymbiosis and the link to prokaryotes. So we'll talk about why there is such a link. And obviously, if you're talking about cell membranes, you need to talk, and extracellular matrix, etc., you need to think about movement of substances across membranes. So why that can occur. So these are the learning outcomes for you. So you should be able to write a good paragraph on each of those, giving examples. And this is quite a good way of preparing yourself for the end of semester exam as well. So have a look at those learning outcomes. Can you actually write about that? And more importantly, can you give examples? Okay? Examples always show us that you know what you're talking about. Now, you've probably learnt by now that rote learning is probably not the best way to approach study. Yes? So always we're going to be looking at, as you go through your courses, we're going to be looking at relationships. So if you're rote learning, you won't be able to make those connections very easily. Okay? So the eukaryote domain, it's very diverse. You've got animals, you've got plants, fungi, protozoa, and algae. So what is, and they fit into this part, so the eukarya of the domains. So the cell structure is very similar between all of those particular species, but you have to be aware that whereas plant cells have cell walls and cell membranes, human cells only have cell membranes. They don't have that rigid structure of plants. But in saying that, they have got a lot of similarities. So you've got a nucleus where the chromosomes, so the genetic material resides, a lot of it. You've got the cytoplasm, that watery environment, plasma membrane, so that surrounds the cell. And there's a number of different inclusions, so organelles such as the endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplasts, etc., that we will be looking at. And hopefully you'll be able to see how important they actually are. So if we go to the nucleus. This is the area where all the genetic material is contained in, and we've got the nucleolus as well. So in the nucleus, it's not there. We have the chromatin, that genetic material, but they can be subdivided as well. So it's not just all the same type. There are different types. And sometimes when you are staining the nucleus or the chromatin, they will have different staining characteristics as well. And you can see that outside that nucleus, you've got a number of different structures that help with processing proteins, etc. The mitochondria producing energy, etc. And you've got some centrioles. These are very important because the eukaryotic cells actually undergo, when they divide, they actually replicate their chromosomes, and the chromosomes, the sister chromatids, actually move to each end of the cell. Okay, and the way they do that is with these centrioles. So it's like an attachment that draws them to either side, to either end of the cell. And then when they have been divided, then the cell will divide in two. Okay, so that's the replication, instead of just binary fission. And we've got lysosomes as well. Lysosomes are quite interesting, and the lysozyme enzyme itself, because lysosome is found in saliva, etc., but also in cells themselves. So if you're a cell and you want to get rid of something, what you can do is combine that with the contents of the lysozyme, and that enzyme breaks down whatever it is in that vacuole, so you can destroy it. Okay, and that's a really good thing, especially when you're dealing with antimicrobial activity. So the nucleus looks like a sun there, doesn't it? So you have the nucleolus that we looked at before. You've got, that is really badly reflecting, and then you've got an inner and an outer membrane as well. So you have got a lot of definition there. And you've also got some nuclear pores and ribosomes that are held on the outside. And you can see these two types of chromatin here, the heterochromatin and the euchromatin, and that's what I was saying will actually stain differently when you, if you are staining those. And so we've got the surround, so the nuclear envelope actually helps contain that chromatin. You've got the pairs of chromosomes, so that DNA material, and they're wound around histones, so they actually condense. And then you've got the nucleolus. This is where you have proteins, DNA and RNA. And sometimes, because you can see these ribosomes, it's the site of ribosome synthesis as well. So there's a lot of things happening in the nucleus. Now, we go from the very centre to the outside when we talk about the plasma membrane. Now, the plasma membrane, as you will remember, is a phospholipid bilayer. And not only has it got those, those phospholipids, it has a lot of proteins, glycolipids. So the proteins can be spanning the whole membrane, or just put into one side of the membrane or the other. And they can be anchors for other sorts of moieties on the outside. So it might be that you have a glycoprotein, so you've got a protein that's in the membrane, and then you've got glucose molecules attached, or lipids attached. So there's a lot of different constructs that identify the cell and help the cell with its activity as well. Now, you'll see in here we've got cholesterol. Cholesterol is very important because it's quite a flat molecule, and when it's inserted in the phospholipid bilayer, it actually moves the tails, the fatty acid tails, apart a little bit. So if there's a molecule that's small enough, it can actually move, and it's lipid soluble, it can move through the phospholipid bilayer, just through the membrane itself. Cholesterol is very important also because it allows that membrane to be to remain fluid, okay? Because it's not just there's the membrane and it's never going to change. It's fluid, so it's going to move. Things are going to be inserted into the membrane. Some things are going to come out of the membrane. Everything's going to be changed around, okay, depending on what that cell actually needs. Now, the cytoplasm is the liquid bit inside, but it's outside the nucleus, okay? It's got lots of substances dissolved in it, and that's where you find the organelles and the cytoskeleton. So these are the things that help with the metabolism and the structure of the cell itself. Then the cytoskeleton, so that the architecture of the cell, you've got a lot of fibres. Okay, so if you think about the cytoskeleton as just being fibres, you've got three different types. You've got the microfilaments. These are the ones that are really quite small, so about 6 nanometers in diameter. And you can see in this image, you've got A is in image A, actin is in red and the nuclei are in blue. So you've got the nuclei here and the actin, so these red bits are part of those microfilaments. Okay, so this is different types of staining. And why are they there? They actually help with movement, so cell movement and also keeping the organelles where they should be, or moving them, or allowing them to move if they need to be moved. Then you've got the intermediate filaments. These are a little bit bigger, and things like keratin that you would have remembered from skin. So these are the intermediate filaments. And in image B, this is from our skin, you've got keratin is in the red and the nucleus are in green. The nuclei are in green. Okay, so once again, different types of staining. Then the microtubules. Now, everyone remembers the flagella, bless you, in motile bacteria. Yes, they are made of, they've got microtubules. Also with sperm, they have got microtubules to aid in movement. Okay, so microtubules themselves are made of tubulin, a protein. They're a lot larger than the intermediate or the microfilaments, and they allow the movement of vesicles through the cells and direct chromosomes to either end. So remember that centriole that I told you about? When the cells are dividing or getting ready to divide, they set out microtubules to attach to the sister chromatids, and they actually help to move the sister chromatids to each end of the cell. And as I said, the structural elements of flagella and cilia, both in bacteria and so that's the prokaryotes, but also in sperm, which is a eukaryote. Then the endoplasmic reticulum. These are quite pretty because they've got these membranous sacs and they surround the nucleus. You've got two different types. You've got the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, or the SER. These, the rough, they're known as rough because they're covered in ribosomes. Okay, that's where you get protein modification and storage. Sometimes you can get synthesis of phospholipids as well. Now, these are quite interesting because everyone remembers insulin? Yes? Insulin is a hormone that aids in glucose metabolism. Yes? So it's not stored as insulin in the cell. It's stored as a pre-hormone, or a pro-insulin or a pre-pro-insulin. Okay? Now, the rough endoplasmic reticulum can help modify that pro-hormone to the active hormone and then export it. Okay, so endoplasmic reticula are very important. Then you've also got the smooth endoplasmic reticulum. This is where you get synthesis of carbohydrates and lipids, etc. And it serves as a calcium depot as well as detoxifying different things in the cell. Now, calcium ion storage is very important because calcium is used as a signalling molecule in basically every signal in our cells. Ribosomes are the ones that help with that protein modification, etc. So you can have, it's quite complex. You've got probably, if you look at the ribosome itself, you've got basically two subunits, a big one and a small one. And they they attach to that mRNA. So you've got, probably if you look at the the image on the right, you've got the large subunit and the small subunit coming together, and you've got this green bit, which is the messenger RNA. And because of those ribosomes bringing it together, or attaching on either side, they can cause or allow the production of polypeptides. And so the polypeptides can be produced, and once you come to a stop, then they can be released. Okay, so you need that ribosomal RNA to be able to produce polypeptides. Now the ribosomes can be bound, so on the rough endoplasmic reticulum, or they can be free in the cytoplasm as well. Eukaryotes have 80S ribosomes composed of a large subunit, which is 60S, and a small subunit, which is 40S. Okay, so, and this is where they're synthesized in the nucleolus. Now we come to the Golgi apparatus, and it is just a series of flat sacs called cisternae, and they're separated by a lumen. Now these modified proteins, so once those polypeptides have been made, they can modify those polypeptides or proteins and lipids from the endoplasmic reticulum by sorting them, tagging them, directing where they're to go in the cell as well. And they can, with the tagging, they can actually put a phosphate group or some other sort of group on them, so that kind of directs them to where they're going. Now in plants, they also have Golgi apparatus, but this is where the synthesis of polysaccharides for the cell wall occurs. So those polysaccharides can be pectin, and you can have 10 to hundreds of these per cell. Bless you. So it's just, it's good to know what they do and what they look like, but it's even better to see what they look like under a scanning electron micrograph. Okay, so you've got 3 different layers. So basically, it's the transport is one way. Okay, so you've got cis to trans, and then you get, so you've got the transport vesicles from the endoplasmic reticulum, then they go this way to form those secretory vesicles. Okay, so it's one-way transport. Now the mitochondria, this is where you get energy being produced. It's enclosed in a double membrane, so once again it's a phospholipid bilayer, and it's got proteins, so it's very similar to the external cell membrane. But what happens inside, you've got a lot of these cristae, so they're in invaginations of that membrane, and this is where you get the cellular respiration occurring. And you can see, like the image here in purple, it will show you where the respiratory enzymes are, and that's going to be important because when we look at respiration a little bit later, you'll be able to have an image of what they look like, where they are. And this is what you would see in an electron micrograph as well. So you've got the cristae, you've got the outer membrane and an inner membrane as well. And these have their own ribosomes and DNA, and there's 1 to thousands per cell. So why would you have thousands? If a cell is really metabolically active, then you need to have those mitochondria to provide, good morning, to provide that energy. Okay, so if you have done anatomy and physiology, yes, you remember in the kidney, in the renal section, you've got cells in the kidney tubules, some that have got lots of mitochondria and some that have very few. If they've got lots of mitochondria, it means that they're metabolically active. They need that energy. Now, in plants, and photosynthetic algae, they have something called chloroplasts. And these are also kind of stacked as well, but they're stacked with these disc-shaped thylakoids. Here, you can see it quite nicely in here, in the image on the left. And there's a lumen between the thylakoids as well. And these stacks are known as granum, and they're surrounded by liquid. Okay, and once again, they also have their own ribosomes and DNA. So they're part of the inclusions inside the plants and photosynthetic algae. So they can use light to carry on photosynthesis to produce energy. Okay, and once again, an electron micrograph showing you how they're stacked. So I think it's quite a nice way to actually visualize those. Now the lysosomes I was talking to you about before, these have got a lipid membrane, but it's only a single layer, and this has got enzymes. Enzymes are contained within that, and they can digest macromolecules. So if there's something that needs to be digested, that vesicle can fuse with a lysosome, and the enzymes will actually degrade what else is in that particular vesicle. And that's quite important, because if you've got a cell that's infected with a bacteria, for example, you can use that. The lysosome can go and basically attack the bacteria or the viruses or other types of antigens, okay, just to get rid of them. Common in animal cells, but in plants it's very rare. Now you can see with phagocytosis, you've got a food particle coming into the cell, and then you've got that food particle in a vacuole. Then the lysosome comes and joins, so these membranes fuse, and then this food particle will actually start to be digested, and then the cell will actually discard what it doesn't need. Okay? Now peroxisomes are a little bit similar but a little bit different. So once again, it is a membrane encased element, but in this case it's got a double membrane. Okay? Once again, it's got enzymes in it, but in this case they're found both in animals and in plants. Okay? And they have a number of different functions. So metabolism of lipids, so you find a lot of these in liver, for example. Oxidation reactions, breaking down fatty acids, etc. And detoxification. So what it does, these peroxisomes, they can neutralize poisons such as alcohol. And what they do is they transform them, or metabolize them, to produce hydrogen peroxide, H2O2. Yeah? And then that H2O2 is converted into water and oxygen by an enzyme known as catalase. And we're going to be using some tests in the pracs looking for catalase production. So you'll know what that actually means. And because it's got those detoxification properties, then you can find, you'll understand why we find a lot of them in the liver. Vacuoles are something that you also find in eukaryotic cells, sometimes in prokaryotes as well, but the space is enclosed by a tonoplast, single layer lipid membrane. Okay, and what that vacuole is, it looks like a bit like a water carrier, doesn't it? So the vacuole is a really good place for storing nutrients and water. So remember the extra large bacteria that we saw last week or the week before? It actually stores nitrites in its vacuole. And because it's so large, sometimes all of the organelles in the cytoplasm are squashed into the cell membrane, okay, to make room for that vacuole. And it's needed to maintain water balance and also gives a bit of structural support for the cell as well. Now the cell wall, this is only found in some eukaryotes, so obviously not in humans, but in the plants, so plant fungal protist cells. These are the ones that have the cell walls that maintain that structural integrity. And what those cell walls are made out of is primarily a polysaccharide. So in plants it will be cellulose, in fungi it will be chitin, in protozoa it's known as a pellicle. Okay, so there's a lot of different names for it, but it's the cell wall. And it helps not only in maintaining the shape, but also in protection. And it's composed of a couple of different layers. So if you think, don't worry too much about figure 3.1 for the moment, but if we look at the image on the right, you can see a cell up the top and down the bottom, okay, that void. So cell 1, cell 2. Now in between those cells, that's where the interesting thing happens. So in between the cells, you've got the middle, which is the middle lamella, okay, between the cells. Then you have the primary wall here on the very outside, and then you have a secondary wall. And that secondary wall has got 3 layers. So you can see when they're butt up against each other, you've got the lamella in between them, then you've got the just outside that lamella, or just deep to that lamella, you've got the primary walls, and then just deep to that, you've got that secondary wall. And the secondary wall has those 3 layers. So it is quite complex because it needs to control movement of substances through the cell or into the cell, and also to maintain shape. What helps to maintain shape in animal cells particularly is what's known as the extracellular matrix. And there's some talk about this being another type of tissue, but that's still talk at the moment. So extracellular matrix in animal cells has a number of different things. You've got carbohydrates in there, you've got different types of proteins, things like collagen and elastins, a proteoglycan network, and glycoproteins, so the fibronectins. Now you probably, hopefully some people who've done anatomy and physiology will remember that the composition of that extracellular matrix is quite important for maintaining elasticity with those fibres, but also with the different types of the proteins, you get a lot more fluid being retained in there. Okay, so it basically is a good reservoir for fluid in the skin, for example, and it holds the cells together. Okay, it helps bind them, so it's kind of like a glue. And you also get cell-to-cell communication through integrins. There's a lot of other types of communication, but the integrins help with that. And also with growth and wound healing, etc. So repair is important. And this table is from Black & Black, and it's a nice one, and I haven't found any mistakes, not like the other one the other week. Prokaryotic cells and eukaryotic cells, genetic structures, intracellular structures and extracellular structures, and how they reproduce. Okay, so it's good to have an overview of what those particular structures are, and particularly ribosomes, etc. Now, if we look at ribosomes, you'll remember that we had the 70S in prokaryotic cells, and eukaryotic cells, you've got an 80S in cytoplasm and on endoplasmic reticulum, but 70S in organelles. Okay, so just keep that in the back of your mind, because we want to know how the 70S organelles got in there. And that was explained by a theory called endosymbiosis, where you have a relationship between two organisms, where one organism lives inside the other organism, or the cell of the other organism. And it's a symbiotic relationship, so there's a positive or a neutral outcome. Okay, it's not a negative one, otherwise it'd be a parasite. So the theory behind that is you get, over time, you get infolding and you get phagocytosis of different, say if it's a eukaryotic cell, you get phagocytosis of a bacteria that produces energy. Yes? Then that's a good thing for the cell itself, the host cell, because it gets the energy from that particular bacteria, for example, prokaryote, and the prokaryote gets protection. So it's a win-win. And that is what's thought to have happened, and that's why we have the 70S in some eukaryotic cells. That's a good thing, except if you're targeting a particular bacteria with, say, an antibiotic that targets 70S ribosomes, because it will also target some of the inclusions in a eukaryotic cell. Okay, so you have to just have that in the back of your mind as well. So why did this theory appear? So about in the 1960s, you've got the evidence, mitochondria and chloroplasts, they're about the same size as bacteria. They have their own DNA. DNA is circular with no proteins, like bacteria. They've got the 70S ribosomes. Protein synthesis is similar to bacteria. They divide by binary fission, so on and on it goes, okay? And bacteria can grow in, or are present in, a number of different situations in cells themselves. So in eukaryotes, in low oxygen environments, they can grow. Some of the protists are found in the hindguts of termites, so they can grow there. And giant tube worms as well have them. Now what I want you to have a look at is, this is, I'm fairly sure this is from the hindgut. But you can see these darker staining areas. They're bacteria. And here you can see them dividing, undergoing binary fission. So it just gives evidence that they can grow and reproduce, so live and reproduce in particular areas. And so we've got all that. Now, we need to provide some way of getting nutrients into the cells or transport things out of the cells. So we've got that phospholipid bilayer. So it has selective permeability, which just means it can let some things in and some things not. Okay, and if you've got a molecule that's very small and it's non-polar and fat soluble, they can dissolve in that phospholipid bilayer and pass straight through the bilayer itself. Okay, but it does depend on being small as well as lipid soluble. If it's charged or it's hydrophilic, i.e. lipophobic, they can't pass through by themselves. They need some sort of transport mechanism. And in bacteria particularly, the plasma membrane is associated with the breakdown of nutrients and the synthesis of energy, so ATP. So there's a number of different things that are happening in those cell membranes. So when we're talking about different types of transport mechanisms, we've got a passive process. So it may be that there's a higher concentration of a substance on one side to the other side. And if it's small enough or there's the right channels, it can move down that concentration gradient with no problem. Okay? Doesn't require energy to do that. And so things like simple diffusion, facilitated diffusion, osmosis. Now osmosis is a special case, and you probably all have done chemistry, so you realise this. Osmosis is the movement of water. Osmosis is water movement. Okay? So that's a passive process. Now the active process, as you can imagine, it means that you need an inclusion of, you need some energy put into the system for movement. Now why would that be? It might be that you need to put one thing from one side of the membrane to the other side of the membrane, but it's against the concentration gradient. Okay? If you think about a bit of physiology here, if you think about your stomach, okay, probably you do think about your stomach, but not in this way. The stomach produces hydrogen ions, okay? Hydrochloric acid is in the lumen of your stomach, yes? The cells actually have to pump those hydrogen ions to a higher concentration, from a low concentration to a higher concentration. So it needs a lot of energy to pump against a concentration gradient. So you need to be aware that that is an active process, and it needs energy input. But sometimes it's nice just to see it in an image. So with simple diffusion, have you ever turned on a gas tap accidentally, and immediately in that immediate area you can smell the gas, but immediately in a remote location you couldn't? Then a few seconds later, maybe more, you can actually smell that gas at that distant location. That's because of simple diffusion. That's all it is. Now, facilitated diffusion is something that is a little bit more complex because you've got proteins in the cell membrane. So we've got a protein carrier. So you've got the phospholipid bilayer here. You've got a transmembrane protein here, and you can see there's 4 sections to it, and it opens up when there's a special signal. It will open up and allow substances to move through. So some of them might just be a straight through channel. Some of them might open up one end and allow something in, and then close that end and open up into the cytoplasm. Okay, so there's a number of different ways those facilitated diffusion can work, but it's basically taking substances from one side and putting them in a regulated fashion into the other side, onto the other side. Okay? Another way, if we're talking about water movement, so this is, passive transport, we talk about osmosis. And typically we talk about, so water's quite a small molecule, and if we had a U-shaped flask or system, and at the bottom of the U we had a semi-permeable membrane. Okay, so on one side we've just got distilled water, purish water, and then on the other side we've got water, but in this case we've got sugar in there as well. So it's a 1% sugar solution. So what's going to happen? Can the sugar go into this distilled water? No, because the semi-permeable membrane won't allow large molecules to move through it. But what it will allow is water to move through. So where is the water going to move? Because if you think about the concentration gradient of water, higher on the right and lower water concentration on the left, agreed? So where's water going to move? it's going to move to the left, to that sugar solution. And you can see that that's, if you look at the levels, that's exactly what happened. Now, why did it stop? Sorry? Equilibrium? Okay, well, if it reached equilibrium, there would be all of the water in here. Gravity, yes. There's gravity, but also, more importantly, is that that pressure of that, known as the hydrostatic pressure. So that pressure that the water, that column of water is exerting. So it's pushing down, the water wants to come back in, but that column of water is so high, it's pushing down and retarding the movement of water. Okay, so that's basically, it's an example of osmotic pressure. Okay, and so, a really good example. But yeah, so you can see the movement of water is osmosis. And if you were to look at the differences between osmosis and tonicity, you've probably heard of isotonic saline or hypertonic solutions, or hyper and hypotonic solutions. Yes, that just means, with that tonicity just means how much the cell membrane actually moves. That's all it is. Isotonic, there might be movement of solutes through the cell wall, but the cell membrane itself won't move. Okay, it won't shrink, it won't explode. So that's a really nice example is if you think about a red blood cell, if you put it in isotonic saline, it won't change shape, so it's isotonic. If it's in a hypotonic solution, it will actually enlarge and probably explode. And a hypertonic solution, it will actually start to shrink, it will crenate. Okay, so tonicity just means the shape of the cell membrane. And as I said, active transport is requires energy, and it also will require typically a protein carrier as well to get from a larger substance from one side of the cell wall membrane to the other side of the cell membrane. And it can also help with modifying substances as well. So once again, requiring energy. So there's a lot of differences between animal cells and plant cells. Agreed? Yeah? And quite a diverse array of those internal organelles as well. The cytoskeleton we looked at, so you've got that structural integrity, that architecture within the cell. The extracellular matrix is important for communication between cells, but also the glue sticking it together, basically, and allowing movement. And the theory of endosymbiosis and why that's good for the cell, but why it might be detrimental in treating, say, a bacterial infection, what sort of implications it could have. So everyone's happy with that? Yeah? Okay, good-o. Some practice exam questions. So, which of the following cellular structures transcribes or produces RNA, ribosomal RNA? Any guesses? B? Okay. What about number 2? A carbohydrate such as glucose is transported across cell membranes in eukaryotes via? Okay, will we have a look? Everyone got their answers? Okay, so we might have a bit of a break and I will come back with the second lecture.